Hi everyone, this is Kevin Wagner, the Keto Advocate. In January of 2016, I had the privilege to sit down and chat with Dr. Laszlo Boros at the first annual conference on nutritional ketosis and metabolic therapeutics held in Tampa, Florida. Let's see what Dr. Boros had to say.
My name is Laszlo Boros. I'm professor of pediatrics at the University of California, Los Angeles. I'm also an investigator for the Translational Research Institute, also at UCLA, and I'm a scientific advisor of SIDMAP, which is a spin-off of UCLA, and we do metabolic tracing using stable isotope tracer carbon labeled substrates as well as deuterium labeled substrates.
Could you tell us about the topic you'll be speaking about here at the conference?
I'm going to deliver a keynote regarding tracer-based metabolomics, or in lay terms, how to trace metabolic reactions and how to follow substrates in a very complex, complicated metabolic network, and how to determine substrate-product relationships. In other words, would an enzymatic reaction take place in a living system, and without identifying or extracting cells of certain populations, we can actually trace these metabolites in the living system as they work, and we can do a real-time assessment of surrogates that are related to certain metabolic reactions. So in other words, we can measure enzyme activity in living organisms and systems and humans without taking anything other than a very small amount of fingertip blood sample. And this is what I'm going to talk about at this meeting, and I'm also going to apply our knowledge and experience in this tracer-based metabolomics to cancer as the most urgent and most sought over area of applying metabolomics or tracer-based metabolomics, but I'm also going to cover sports medicine, individual variations on individual variations in metabolism, and also drug response and drug target research. We do a number of different areas of research that cover different disease processes. One is a disease process called Huntington's disease, which is a neurological disease, and we are specifically determining certain metabolic abnormalities that are associated with this disease and how to overcome these abnormalities using ketogenic diets. And we are looking at the biomarkers as well as the efficacy biological, biochemical endpoints as far as 13C labeled products in certain brain areas and also in plasma of animals. The other main area that we cover is cancer metabolism, and we do have really broad collaborations among universities that include the University of Arizona, hospitals from Europe, and we are actually exploring a very new area of research that is called deuterium depletion, and this relates to mitochondrial metabolism, because the new information that we gathered over the last few years is that hydrogen, which is a very essential part of all structural molecules, for example, DNA, is linked with hydrogen bonds. People probably recall that there's a double helix of sugar molecules, and there are certain bases attached to those, and these bases are held together by hydrogen. And if this hydrogen is replaced with one of its stable isotopes called deuterium, because deuterium has twice the mass of hydrogen, there's going to be a huge epigenetic event which has not been focusing or not been explored for cancer research. And for example, DNA repair enzymes would stop when you bump into a deuterium. It's pretty much like when you cut your grass in the backyard and there's a big pile of grass that is exceeding the capacity of your lawnmower, then the whole system dies. So that's how DNA repair is really dependent on deuterium hydrogen ratio in DNA. And it seems that this is regulated by diet, because natural fat has low deuterium, so the mitochondria and the mitochondrial beta oxidation process produces water that is deuterium depleted. And this water gets into DNA or affects DNA deuteration, which actually is an epigenetic event. So as the novelty and some of the topics that are leading into submolecular applications in cancer or explaining cancer beyond genes or the level of genes, we learned that actually hydrogen deuterium ratios, because of the large kinetic isotope effects of hydrogen versus deuterium, or in other words, deuterium versus hydrogen, have major biological impacts. And just let me give you an example. If you look at a water molecule that has an oxygen-hydrogen bond, if you replace hydrogen with deuterium, it's going to take 7 times longer to break that bond, and it would take significantly more energy to break that bond. And that's the same in hydrogen bonding networks in biological systems. So the deuteration of our cells or DNA and its fat compartments have a major impact of their phenotype or biological behavior. And actually diet controls this process. Usually sugars or carbohydrates are high in deuterium, and on the other hand, fat and ketogenic products are low in deuterium. So when we oxidize these substrates, our mitochondria, in the first step, they always produce water. There's different deuterium content in mitochondrial matrix water, depending on what you oxidize. And ketogenic diet, we believe, contributes significantly to the intracellular, more specifically intramitochondrial deuterium depletion, where this water is produced. And therapeutic effect can be mimicked using deuterium depleted water as drinking water as a very efficacious cancer treatment. In very aggressive cancers, we can actually make patients who drink deuterium depleted water in coordination with their standard therapy sometimes 4-5 times longer than they would just have survival on standard medical care. So obviously, understanding the biochemistry of it is a major area to explore, and that's the main topic of what I'm covering at this meeting. I heard talks, and I talked to investigators where it seems that other beneficial findings or improving cancer treatment or improving survival can be put in the context of the biochemical mechanisms that we have clarified. So we have a lot of things to talk about. We have a lot of dots to connect with these excellent investigators, and a biochemist like myself can actually always be part of the conversation and be always part of the discussions and interpretations of how certain therapies or how certain alternative therapies or how certain adjuvant therapies work, and how to develop certain markers or biological endpoints or tracer-based metabolomics substrates that can be followed very specifically in metabolic networks, how those can be applied to get closer to their mechanisms, because that's always key in medicine and in biomedical research, to find a mechanism that can be consistently applied to various systems and enhance knowledge and give that knowledge to the hands of clinicians. Because we are in the background in the sense that we don't treat patients, we don't advise medical treatments, but educating our physicians and giving tools in their hands without necessarily all the scientific details. Yet, from the scientific point of view, it's solid and definite. That's our responsibility to clarify all the risks and hazards that may be involved in certain treatments, to keep this conversation and make this conversation part of the translational research efforts. That means carry scientific discoveries to the bedside is what we follow through during our research efforts. And this is why I'm so thankful to be part of this conference, being invited and given the opportunity to talk about my research. So UCLA has websites where faculty is listed. I have delivered a number of talks to medical students, and also actually UCLA is asking for a course proposal where we teach deuterium, which is deuterium biochemical processes that alter or control deuterium hydrogen ratios in our biomolecules, and it's offered for the undergraduate honor class, so or for credit. So it seems that the university is very open-minded, even from the educational point of view, to turn directions towards metabolic biochemical patterns instead of genomics research, and let us educate students beyond the genomic kind of era or downstream of the genomic events, using very specific, very powerful metabolomics technologies. And the best source of information is the medical literature, where we publish our peer-reviewed medical papers and data. And I have published some close to 90 papers in the medical peer-reviewed papers in the medical literature. ResearchGate is a good source of papers and interpretations and talks, presentations, and unpublished data and raw data, where actually anyone can follow a certain scientist or researcher and compare their peer review process in a very broad community, because there are more than 8 million scientists who actually make up this ResearchGate scientific community. And obviously more thoroughly and publicly anyone can follow certain talks on YouTube. And my YouTube channel is fumarate hydratase, which is an enzyme, metabolic enzyme, which was the function of it was described by Dr. Albert Szent-Györgyi, who got the Nobel Prize in Medicine in 1937. And he happened to be a friend of my grandparents in Hungary, and my grandma talked about their family relationship, and they actually rented the same season ticket in the Szeged Opera, because that's where he was actually a faculty member. And best of my childhood memories as far as science goes, grandma talking about this Nobel Prize winning biochemist, and I think that was the bug that eventually infected me to be a biochemical knowledge for the benefit of mankind through clinical applications. Metabolic tracing is following specific substrates and determining the production of certain products in a very targeted, very directed manner. And those, most of the time, include biologically relevant reactions in a very complex metabolic network. It's pretty much like highlighting a map route just to follow certain directions and just being interested, finding out what's the traffic in that particular venue. And that's what biochemistry or tracing biochemical reactions would do, is using a unique glucose molecule, for example, that has carbons that are actually heavier than the natural carbons. Then based on this weight change in products, we can determine the rate of synthesis and the rate of breakdown. We call it turnover. And we can actually go very distant in a biochemical network given enough time to these substrates to kind of populate or enter different product pools. And based on this information of what time and what substrate concentration would label what product pool, and how that change over drug treatment or cancers like cell transformation, that gives us specific information regarding the metabolic network, how it is involved, through what branching points there are substrates acquired or accumulated by tumor cells to be able to grow in an unlimited, aggressive fashion. And this is what metabolic tracing does. It actually finds those very specific enzymatic reactions which are responsible for this growth by labeling the substrates that go through those pathways. It's pretty much like you want to determine your city traffic, and you use a few cars that you paint them in a certain color, and you follow those cars, and you determine what way they get from point A to point B. And it's very useful information when there are alternative routes or there are alternative ways to get to certain points. Then you can eventually, if you let these cars circulate long enough, you can actually decode a very complex metabolic network or a traffic situation and find out what the tricks are that tumor cells are using to be able to grow in an unlimited fashion. So let's put it this way. The carbon molecule C, carbon, has a nuclear structure, has 6 protons and 6 neutrons. So it adds up to 12, and that's the mass number of carbon. And this is 99% roughly of all carbons that we have on our planet. Now 1% of carbons have 1 extra neutron in the nuclei. So it has 6 protons and 7 neutrons. So it makes up 13 particles in the atomic nuclei, which makes this carbon 1 dalton heavier, or 1 mass unit, atomic mass unit heavier than the rest of the carbons. And this is 1.1% of all carbons, just like as we said here, all the CO2 that we breathe in and out, actually 1.1% carries this heavy stable isotope. It's stable, not radiating, so it's harmless. But there are certain ways of collecting these carbons, and if we actually provide these carbons as the source of molecule synthesis during photosynthesis, we can actually grow plants that build their structure based just containing nothing else but this 13C carbon. It's an expensive process, but for biomedical use is very helpful, because we can generate glucose, for example, that these plants produce when their source of carbon is only 13C. That glucose that has 13C carbon substituted for the 12C carbons, which is the natural most common form of carbon, and we can actually apply this heavy carbon containing glucose to cell cultures or animals or humans, and we can now trace the fate of this carbon through metabolism and recover it and the fragments of it, or the biochemical products of it, from many other metabolites or products that our body would synthesize over a period of time. Now, looking at the possible scenarios, how these products can label plasma metabolites, we end up with a huge database simply because we're not only looking at metabolite concentrations, but we also look at their C12, which come from other sources than the labeled glucose, and C13, which come from the labeled glucose over a period of time. So we need to run a mathematical model, or we have to approach this very large data set using mathematical tools to actually find correlations or associations, or using, it's called regression statistics. We look at how, for example, labeled glucose disappear in the plasma over a period of time. It's usually 30, 60, and 90 minutes. And in the meantime, we measure how much fatty acid gets labeled from this glucose, and we do a correlation statistics. And if there's a cutoff correlation value, either the positive or negative direction, is the fatty acid labeling or cholesterol labeling goes hand in hand with the disappearance of glucose, then we can establish that certain enzyme activities are producing products from the labeled glucose, and these association patterns will give us an idea of what individual variations there are in metabolism, what enzymes are active, and truly what drugs should be applied to treat certain diseases, simply because we know now the activity of these drug targets. And so we can actually use this for personalized medicine, and we can also position drugs or actually treat patients, not only statistical evidence that is now so common in medicine, like evidence-based medicine, but now we can bring this information down to the individual. So we don't have to follow certain statistical trends, like this drug would work or not. We don't have to take this as a chance anymore. We can actually answer the very specific question to help the physician: is that patient would benefit, or would that patient benefit from a certain drug because certain biochemical pathways are in place, because now we can measure virtually almost all biochemical reactions that take place in our body. So tracers have been around, and for the biochemical applications for different disease processes, they started using them like from the late 60s. But actually, those were more like radiating isotope type of work. The first solid applications of 13C labeled substrates in biochemical systems started probably in mid-80s, when mass spectrometers, which actually can sense weight difference or mass difference between biomolecules, were in reach of scientists. And one scientist that I started developing certain disease applications is Dr. Paul Lee at UCLA, who solved many of the mathematical problems and the distribution statistics and patterns that need to be done before we would interpret any labeling data, because these isotopes occur in nature. So there's always a background subtraction issue. And my contribution to the field was applying these tracer technologies to cancer. So basic biochemical information was collected by many investigators, including Dr. Henry Brunengraber. I don't want to leave out anybody, so I don't want to list too many names, but there are many outstanding investigators who cleared the way for tracer-based metabolomics and data interpretations. My particular contribution was to apply this technology to cancer, and in collaboration to obesity. And we actually clarified a few biochemical phenomena that were disputed at that time. So the tracers, especially glucose tracers, allowed to measure glycolysis and the pentose cycle, which is affiliated or associated with glycolysis, but it's a kind of a side way of processing glucose, and in the meantime, there are certain cofactors that are benefiting tumor cells. And using this glucose, we could actually very precisely tell and kind of predict how tumor cells behave or use their metabolism differently than normal cells. And because of this information, we could actually develop certain drug targets that seemed promising. And continuing on that line, this is how we ended up in looking into mitochondrial metabolism, looking into ketogenic diets, because they all have their impact on metabolism one way or another. And we can now correlate these with anti-proliferative effect, anti-proliferative mechanisms. For example, we can explain how chemotherapy and how radiation therapy work together with ketogenic diets. It's been a long way. Obviously, it took 20 years to get to some very solid clinical applications and mechanistic understanding of certain processes. A lot of data was collected in the meantime, and they all had to be interpreted and reinterpreted and eventually incorporated into certain drug development efforts and biomarker development efforts, as well as toxicity markers, or understanding in general cancer epidemics or increasing cancer rates or failing treatments. These are all part of this, I would say now, very robust effort of using metabolomics and tracer-based metabolomics to tackle some cancer-related issues that would not be tackled using any other technology. So there are certain depths of biochemistry, there are certain depths of functional biochemistry and downstream effects of gene actions that cannot be understood without tracer-based metabolomics, or tracers that are actually followed and traced through metabolism. And this is the type of information, I think, which will make a huge impact in the clinical assessment and clinical management of cancer for the future after so many targeted therapies are failing. We label substrates, stearic acid and palmitic acid, which are fatty acids. So we didn't label ketone bodies, but we labeled fatty acid products. And those get into cells, those get into RNA and DNA, and those are actually part of the metabolic water production process in mitochondria, because surprisingly, there's water that is produced in our body. Actually, not many species consume water on our planet. Certainly no desert animals consume water. Interestingly, fish doesn't even drink water. They produce their own water. And if you eat fish meat, it's not salty at all. So their water is produced from metabolic water. It's like the camel. Camel don't drink, and they go through the desert practically without drinking. It's because of the fat in their bumps. So as long as you can oxidize fat and there is oxygen, and that's where hyperbaric oxygen treatment comes in, you can burn as much fat as you want, and that will give you water. ATP synthesis is a different story. Energy production only comes in when you recycle this water and keep producing ATP. Obviously, it will give you a metabolic advantage of you can actually populate the temperate climate, and you don't have to hibernate at night, just like desert animals. The desert animals have to hibernate at night because they use their water for their body solvent capacity, use water as solvent, and they don't really recycle it through the TCA cycle and keep producing energy. So that's why they have to use the sunlight to generate heat. So there's a big gap between hot-blooded or mammalians and desert animals, simply because they use water differently. different rate of cancer or a different ability to develop cancer between and among these species, and understanding this biochemical diversity and clinical diversity of what species can, if they can develop cancer in that malignant term, that actually understanding this would be very important as far as understanding deuterium and hydrogen and water production. PEPCK is a phosphoenol pyruvate carboxykinase. I know it's even more scary than the abbreviation, but what it does is it allows our cells, it's an irreversible reaction in biochemistry, meaning that it never goes back, meaning that the product can never turn using the same enzymatic reaction back into the substrate. It would take another metabolic set of reactions to break down the product in a different, through different steps. These are what we call irreversible reactions. There are many reactions that are reversible, meaning that they can go both ways, but this PEPCK reaction is irreversible. So it produces actually, in very late terms, eventually a neogenic, so a substrate that can produce glucose in our body or in our cells. And tumor cells, they love glucose simply because it's the ready source of nucleic acid synthesis, and it's also the source of energy, making energy in a very sluggish way. But if there's a lot of substrates, then, and they can build substrates, they can actually use and utilize this pathway. And this particular enzyme is responsible for restoring glucose in tumor cells using substrates that are really not like the sign are prone to develop glucose or form glucose in normal cells. So it's an enzyme that some tumor cells depend on to grow, because they can use it to form sugar or glucose. And this is why it's so important, because if we understand or we can actually target this enzyme, those tumors that actually depend on it likely will have to cope with the situation that they cannot really produce for themselves, so they have to withdraw from the cell cycle, from the division cycle, or die, or adopt a different substrate environment, or use different enzymes, which we can again trace with tracers, and we can catch them again. So tracers would allow to find that enzyme, determine that enzyme activity in tumor cells, and find out if the patient would benefit from an inhibitor of this enzyme. And this is why this critical enzyme with this critical tracer can be a good tool for the clinical management of certain cancers that include colon cancer and some very aggressive gastrointestinal cancers. And this is, in a nutshell, this is what this enzyme does. Probably there is, but again, PEPCK is active in certain cancers. We have to find those first. And if that's the case, then PEPCK has a therapeutic benefit if we regulate it. But it's not a general thesis, or it's not a general approach. So, and even metabolism, we have to be very careful with metabolism, because actually cancer cells find very quickly some alternative ways. So of producing, for example, glucose, which they would use this enzyme for, using substrates that are coming from amino acid metabolism and so on. So first, we need to find those tumor cells that actually depend on this enzyme, and then we can have a good chance of clinically interfering with their growth activity, simply just modulating this enzyme. But we always have to be ready for what we call therapy resistance or drug resistance, where actually we have to switch our strategies because specific inhibitors of certain enzymes or proteins may not work for a very long time, and resistance may develop, and they offer benefits, but those benefits are only temporary. So yes, indeed, but we have to adopt this in a very targeted manner. So the source, well, first of all, fructose is a sugar, and it comes from fruits. It's called fruit sugar, and it has the same, almost would say, the same composition as glucose, but the hydrogens and oxygens are a little bit rearranged. And fructose is the product of glycolysis that actually glycolysis product, but it's partial metabolism, meaning that certain tissues produce or use fructose very intensely, and some others don't. Practically, it's a substrate that is not regulated by our liver, but it can come from sweetened drinks, carbohydrates, fruits, especially fruits that are grown in the fall, and it's part of soft drinks. Levels can go as high as almost like 4 or 5 times more or higher than glucose based on the drinks, based on these sugar drinks. And since the U.S. diet, or because of fructose is a common sweetener, it costs less, and it's 10 times sweeter than glucose, and it can be produced from corn. It's a very common substitute or sweetener in Coca-Cola and some other food products. So pancreatic cancer patients, based on their diet, or any cancer patient who is in a sugar chunky diet, they actually will have very high circulating levels of fructose in their plasma. And fructose is, as far as metabolism goes, has different values than glucose has. And fructose is basically a sugar that will make liver cells to produce fat. And fructose also compromises glucose oxidation or breakdown. Fructose also compromises fatty acid oxidation and breakdown. And it's also part of nucleic acid synthesis by providing a certain part of its skeleton to nucleic acids, especially newly formed RNA and DNA. So in that's called a enzymatic reaction called transkylase. So fructose as a substrate is really not desirable to be part of a cancer patient's nutrition, simply because it's the ready substrate for nucleic acid synthesis and fat synthesis. It's an anabolic, this is how we call it. It's an anabolic sugar, so it doesn't get broken down. It actually builds other molecules that are included in fat storage processes and also nucleic acid synthesis. So understanding these mechanisms more clearly is where this fructose or sugar positioning, or understanding sugar metabolism was so important to us, and this is why we linked with most aggressive cancers like pancreatic cancer. It's because fructose gives part of its skeleton to nucleic acids, and the enzyme that is responsible for that is transkylase. So the substrate and enzyme, when they are in place, tumor cells are very happy to use these very simple biochemical reactions to build new nucleic acids. In other terms, to be able to proliferate and accomplish their uncontrolled, unlimited growth. And if the substrate environment is optimal in that sense, if there's high fructose circulating in plasma or there's high fructose in tissues, then they can take advantage of that. And the key enzyme to that process is this transkylase. And we call it transkylase because it transfers part of fructose to another substrate that would accept it, that come from fatty acid breakdown. It's called glycerol. It's a very common intermediate metabolite. But it seems that tumor cells can have access to fat or adipose or triglycerides from circulating triglyceride pools, and they can actually hijack the two top carbons of fructose or part of the fructose skeleton and synthesize nucleic acid right on the spot. So it's a very efficient way of synthesizing nucleic acids, and fructose is a good substrate for it. In general, cancer patients, as all other, like even healthy populations, they have to stay away from excessive vitamin supplements. And the reason for that is vitamins can work like two ways. They support and they are necessary for normal cell functions, and their depletion is sometimes life-threatening. But oversupplying vitamins can also trigger and obviously can support the growth process of tumor cells. So excessive vitamin supplementation, and I'm not just talking about vitamin B1 or thiamine, but excessive vitamin supplementation should be looked at very carefully, simply because cancer rates are going up. We know now it has to be some kind of a nutritional or biochemical process. It's not a genetic disease. We know now it has to be some kind of a nutritional or biochemical process. It's not a genetic disease because there's really no consistent genomic or genetic findings behind cancer. Now there are over 100,000 genetic alterations or mutations in tumor cells, which really show no clear patterns. And obviously 20 years of genomic research just resulted in nothing as far as being able to diagnose or treat cancer patients. There are certain treatments that drug companies claim that they have effect, but sometimes it's because of the toxicities and the hospitalization of the patients that actually make them live longer. So it is very marginal, like statistical findings, like patients would live like 4 weeks longer. The average is, I think, 6 weeks longer in clinical trials with these targeted genomic signaling pathway drugs. Is really not the discovery or not the understanding of these pathways, or it's really not very impressive as far as treatment or efficacy goes. Their price is impressive. I mean, some of the cancer drugs are over $100,000 a year. Actually, the newest one is over $1 million now. If you compare the price of this drug to the price of gold per grams, actually this cancer drug is 4,000 times the price of gold and practically has no significant effect. So we need to be very, I mean, we need to go back to the blueprints, and we need to kind of find out what went wrong. Like war on cancer, I think President Obama just announced his second war in that sense, just as challenging as landing on the moon. They have to include some alternative approaches other than genomics, because if genomics didn't work in 25 years, let's say just like since 1990s, since NCI started funding nothing, practically just genomic research, if it didn't work, more and more patients die every day, more patients die every year of cancer. You can dispute if the rate of that is like what the kind of the slope of that, but actually it's more people every year. So to me, slope doesn't matter. We need to employ alternative and more efficient metabolic mechanisms and approaches to be able to get to the bottom of this problem and resolve this very scary social trend or disease trend, which is in Western countries, cancer is the leading cause of death. Every third person over the age of 65 will develop some sort of cancer. And these statistics truly alarming the scientific community that we have to come up with some alternative and hopefully better ideas than cancer is a genetic disease. In my reading, cancer is a metabolic disease affecting cells. It's not a metabolic disease like we would think of like diabetes or obesity. No, it's a cellular metabolic disease. So there's metabolically something terribly there's terribly something wrong. Well, we know what is wrong with metabolism. It's practically there are certain deuterium overloading processes that can actually make our DNA heavier and act as a epigenetic event, as a continuous growth signal. So ketogenic diet, mitochondrial functions, metabolic water production, deuterium depletion, deuterium depleted water consumption, actually less water consumption and making water produced in our body using fat or fatty diets, which are deuterium depleted. That's the natural way of regulating our metabolism and DNA homeostasis, and this is what we are kind of exploring of how to use this information for medicine. This is actually a very interesting story in that sense that it goes back to the 1800s. There's this Belgium, I think he was Belgium scientist, Lobry de Boon, who discovered that actually turning glucose into fructose or turning fructose into glucose through an enzyme called isomerase. Well, at that time they didn't know about enzymes, they just knew about this chemical reaction of what we call isomerization, when actually you don't change the chemical structure other than that you just rearrange certain atoms in the molecule, and that's what glucose and fructose does. It actually you switch hydrogens and oxygens between two carbons, or at least in a more broad terms, the first three carbons are involved in this hydrogen-oxygen exchange. Now, for a very long time, and still biochemists think that actually this hydrogen switch takes place in and within the glucose and fructose molecules. That's not true. Actually, the hydrogens always get picked up from water, from cytoplasmic water. And hydrogen is consumed, but it's not the same hydrogen that jumps from one carbon to the other, but there's water incorporated in the process. So this water, whatever is in the cytoplasm, will determine the hydrogen content of fructose and not the hydrogen content of glucose. So there's this water, like the intermediary guy, that actually regulates the deuterium content of fructose. And since fructose is the source of DNA, it can deuterate DNA unless if the deuterium comes from glucose. But if there's deuterium depleted water in our cells, the heavy deuterium on glucose can be switched to a light hydrogen when it turns into fructose. So providing deuterium depleted water as the source of water for this reaction, we can regulate deuterium content in our DNA sugar backbone. In that sense, we can actually make our DNA more sensitive to breaking by radiation therapy. And we can actually regulate the amount of DNA based on this deuterium depleted water and hydrogen deuterium switch between glucose and fructose when this isomerization or this reaction takes place. In very scary terms, it's called the low bilirubin aldose ketose transformation. That's just to scare you guys, but practically that's the exact biochemical term. And it uses water as an intermediary substrate. So deuterium depleted water in our cells can actually deteriorate glucose by the time it gets to fructose. And that's what green plants do. So green plants, it's very surprising, if you isolate glucose from beans or from spinach, actually it has half of the deuterium on our second carbon, just like glucose would. And it's simply because of this reaction. So this reaction is a natural deuterium depleting process, and this needs to be explored as a therapeutic potential to enhance radiation therapy or the efficacy of radiation therapy, chemotherapies, or in general to control DNA deuteration and the epigenetic events of deuterium, which is again has and possesses very strong kinetic isotope effects, meaning that it's really like twice as heavy like 8-ball. It's really deep, you know, when you use a pool game, all those balls have to be the same weight. Once deuterium gets into the story, it's like you actually put a twice as heavy 8-ball in your pool table, and all those hydrogens, they can bump into it, but it's not going to move. And that's what deuterium does in metabolism. Usually, I describe this to my students as these cowboys, like the Once Upon a Time in the West when Cheyenne came into this pub in the beginning of the movie and all the horses stopped breathing. That's practically what deuterium does to metabolism. It's actually the bad guy. There's not many of them. There's only one in 150. There's only every 6,000 water molecule would carry 2 deuteriums, and every 3,000, 1 out of 3,000, roughly 3,000 water molecule carries 1 deuterium. But because of its magnified kinetic isotope effect, eventually even I usually tell them that every deuterium counts. It's like every bad outlaw in like New Mexico had the entire state on edge simply because that's how bad they were. And that's the truth of deuterium. So we have to control it through very tight, very precise biochemical reactions. And this takes us to the submolecular level of cancer development or oncogenesis, which was actually suggested by Dr. Albert Sangiorgi back in the 70s, right after Dr. Warburg, who described the Warburg effect. So metabolic therapies, well, actually, Dr. Sangiorgi was talking about electron transport, which is part of this hydrogen transfer process. And but actually, the initial ideas come from very respected and very bright Nobel Prize-winning scientists. It just seems like genomics took over with not much benefit, but obviously we are trying to get back on the same horse and see if there's still life in it, and you know, let them pull our cage much farther than any other options there are. I think that's the next very logical step, and it's going to be discussed at this conference after my talk this afternoon, because I'm going to offer some methods and practices how to use this and on a very routine basis, meaning that it's really not that magical. It just has to be interpreted using solid mathematical and medical information, but technically it's really not that difficult. Yeah, like as a kid, like I can't imagine just like anyone would be mesmerized by a story like that, because my grandparents, they lived on the same floor. They even like rented the like they were housed in the same apartment complex back then. And Dr. Sanjay's wife was talking about his obsession with paprika, where he isolated vitamin C eventually. And he was actually, he wasn't eating his paprika. His wife offered him for dinner. He always put it in his pocket, and he took it in his lab. And he was extracted. He had to take some over 2 kilograms of paprika over 3 months to actually isolate 1 grams of vitamin C from it.
He didn't exactly pick the most concentrated raw material, did he?
Yeah, right.
Yeah, because as he described it, because it tasted so good. Because it tasted so good, and he figured that there must be a good source of a unique chemical that like works as a vitamin, and that's how he isolated vitamin C.